Hello everyone, welcome to today's live broadcast, Small Refinements Equal Large Enhancements to Science and Animal Welfare. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals. Before we start, there are a few instructions. This is an educational webinar and thus offers free RACE continuing education credits. After the webinar is over, and to get your CE credits, click on the CE button located in the bottom left-hand corner of your web page. And we want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answer is welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Letty V. Medina, DVM, Diplomate of the American College of Laboratory Animal Medicine and Director of Animal Welfare and Compliance for AbbVie Incorporated. Dr. Medina earned a Bachelor's of Science in Biology at the University of Notre Dame and a DVM degree from Texas A&M University. She completed her laboratory animal medicine postdoctoral training at the University of Illinois in Chicago. Dr. Medina serves as the Director of Animal Research and Compliance at AbbVie. Her primary responsibilities are to ensure that AbbVie adheres to all animal regulations, adopts the most ethical animal research practices, and continues to enhance an ethical culture for animal research with innovative 3Rs initiatives. She has served as chair of the 3Rs leadership group of the Innovation and Quality in Drug Development Consortium in 2014 and as a co-editor for the 3Rs Special Topic Journal published by JLAS in March 2015. Dr. Medina is dedicated to promoting more transparency about ethical animal research to ensure public understanding and support for biomedical research. She is a frequent speaker at national meetings about animal welfare, the 3Rs, and the need for public outreach. I will now turn it over to Dr. Medina for her presentation.
Good afternoon, everyone. I think the people I'll be presenting to are in the afternoon, hopefully. So thank you for attending this talk. I'm going to just go through kind of an overview of what I call small refinements, which can lead to large enhancements to science and welfare. So I like to look at it as a holistic approach to the way we do work, starting with the way we transport the animals and receive them into our facilities, how we acclimate them, and then all the way through the process of caring for them, enriching their lives, the way we handle them, and different oversight procedures, as well as trying to advance the 3Rs whenever feasible. So when we talk about optimizing animal care and use practices, we have to think in a holistic manner. So it's not just the animal care staff that are caring for the animals every day. It's not just the veterinarians. It's everybody. The researchers and the IACUC staff, they all have an important role to play to ensure that our practices are optimized and to reduce unnecessary pain and distress. And so if we strive for uniformity and best animal care practices, we have consistent humane animal study techniques, and we really strive to adopt the 3Rs when they're feasible, then I think, again, we're all working together to optimize that animal care and use. Please, just a reminder that anybody can ask a question in the middle of the talk, and I'll try to stop and answer if you have any questions. So starting at the beginning with transportation, how do we optimize transportation to enhance the welfare of our animals? So, I don't know how many of you take the time to review the transportation log, the food, the water, how the animal's been observed if it's a long transport that they've come, from a very far distance to your facility. And it's our obligation, in a sense, to point out gaps if our transportation companies maybe, aren't doing everything to our specifications. So we have to work with them to optimize the care of the animals. Things to consider, like for rabbits, we've learned this in our own facility, that when possible, because ALAC is now emphasizing, pair housing or group housing of most species as the default. For rabbits that tend to fight quite a bit, we've had success ordering younger siblings when we have to bring in more than one rabbit at a time, and to transport them in pairs so that they can remain together and then you have less fighting. We also try to get immediate weights on those rabbits, since they tend to be a little stressed from the transport and they can lose weight, that first week as they're getting acclimated to a new environment. Another trick to kind of keep in mind to work with your transport companies is if there's stormy weather happening, do they use white noise or music to kind of calm the animals so that they're not quite so startled by loud thunder and things like that. And then just a log of adverse events. A few times we've had our comparative medicine group come and talk to the IACUC about some incidents that have occurred, some welfare issues that were not handled optimally by the transport company. And so again, then the IACUC will work with the transport company to make sure that we're reducing the likelihood that that kind of adverse event will happen again when we're receiving animals. With regards to receipt of animals, how do we receive them? So with dogs, we've come up with a pretty interesting way to do this by actually allowing volunteers who don't work in the animal program day in, day out, but who just love dogs. They come and volunteer with our animal care staff to help pet the dogs and interact with the dogs, because the dogs are a little nervous when they first arrive. And it's a wonderful way to get those dogs, brought in a more optimal way. I know that's maybe not feasible for all the programs that are out there, but it's been a real success here at AbbVie, and I would certainly encourage you to consider it. I'll talk a little bit more about that kind of thing later. Another thing, with non-human primates, if you're working with non-human primates, just making sure that you're looking at the food in the crates and looking at their feces. Do any of them have diarrhea? Are they, possibly going into a state of not wanting to eat because of the stress? Just so you can be prepared to, keep a better eye on them when they come into the facility. And then just to optimize the arrival and receipt of surgically altered animals, having veterinary staff there to kind of look at the incisions as we're encrating them and making sure that if there are any, maybe some, open wounds or things that have gotten, a little bit traumatized during the transport, that we deal with them in a very prompt manner. So just things to keep in mind, is your IACUC informed when arriving animals experience health or other complications? I do think that is something that we should make sure that we do let our IACUCs know, so that they can help to manage that if it needs to be addressed in any form or fashion. And then just again, shipping those pairs when you can with the rabbits, and then reviewing the process for all the species, because they're all going to have different transportation needs, and making sure that we're partnering with the transportation companies to optimize things as best as possible. Okay, so when it comes to the next thing, acclimation. So obviously in the guide, it's very clear, we all know that newly received animals should be given a period for physiologic, behavioral, and nutritional acclimation. Animals typically need a minimum of 24 hours for rodents and 72 hours for larger animals. Obviously, some programs will establish more of the standard 72 hours for all species. A 3-day acclimation period is not uncommon. But I think it's very clear that acclimation has been demonstrated for a very large number of species. These are some of the references in the guide. And make sure that you really have a pretty consistent way of dealing with that. So you don't have animals coming in and going straight on study, which isn't optimal for the science or the welfare. So some of the considerations, again, do you have an SOP for each species or for all the species in one SOP, however you want to deal with that. One of the examples that I'll give from my program is that we put rabbits on a feeding schedule. We have a sticker that we put on their cage cards as soon as they come in, just so that because they're given a slow acclimation to going on increasing feed ration over several days, so that they can kind of manage the stress and their response to maybe not eating as much, and they don't get upset stomachs when they come in. And making sure, again, that your IACUC is in on the loop and reviewing those kind of guidelines as new information and science is done on these different areas of, the impact of stress on animals. Moving on to husbandry, some really important things to consider with food. Very recently, many of you may already know this, and maybe you've dealt with this in your own program, but we just recently, in the last several months, have been dealing with this a little more acutely, and that is because we're a pharmaceutical company and we have animal vivariums at several different places around the globe, both in the United States and outside of the U.S. What we've realized is when we were doing similar studies in one site to another site, that we were having some different data outcomes, as something as simple as the drug levels we were measuring. And when we looked at the comparison of how we housed them and how we managed them, it came down to the food, and the soy-based diets were affecting a difference, making a difference in the data we collected. So we recently saw plasma concentrations reduced in mice on soy diets. And the theory is that the soy induces the CYP enzymes in the liver, leading to faster metabolism of the drugs. And obviously that can be a real big impact on pharmaceutical research because we want to know what the plasma levels are and how they're likely to translate into human clinical trials. You might want to look at this reference that I put up there from Westmark from 2013, even saying soy-based diets exacerbate seizures in mouse models. So we have to really be thinking about the bigger picture for these things and how they impact the science. Here's some of the data that we collected internally, that when we compared soybean meal in red with corn gluten meal added to normalize the amount of protein, the animals on soybean still had lower plasma concentrations of the drug compared to the animals on the corn gluten meal, the non-soy diets. So again, please keep that in mind as you oversee your individual programs. Other things to consider from a welfare perspective with food is just caloric intake. Obviously, this is something that I think we all have to deal with all the different species we handle, especially if you have chronically housed animals. I've certainly seen problems with obesity in rabbits, in rats, in dogs, and in monkeys. I've seen it in other species too, but these 4 that I have listed on the slide are the ones that I've had the most concern with, because obviously with rabbits and rats, the obesity can lead to foot problems if they're housed long term. With the monkeys and dogs, you can get metabolic syndrome or type 2 diabetes. All of these obviously are not optimal for welfare, and they can affect the science and the research that's being done. And so if we can control the feed intake and limit it, the animals are maintaining much better body weights. And I think, again, it's something that sometimes we do a great job, and depending on the size of your program, it might be a little bit more challenging in some of our programs. Something as simple as bedding. So a number of years ago, back in 2011 and 12, our comparative medicine animal care staff group decided that they were going to change bedding from aspen chip bedding to corncob bedding, because the corncob bedding was going to save money and it was going to be more absorbent, and they thought that it was going to be better for the animals. Well, lo and behold, several of our research staff quickly came to the animal care group and started saying our research data is not working right now since we made the change to this corncob bedding. And so you'll see that this one publication by Laura Lays and Steve McGarity, that the rats housed on corncob bedding show less slow wave sleep. And we published that in JLAS back in 2012. And if you look at the red arrow, the low amplitude waves from the EEG rats show that they're awake. So the rats on the corncob bedding in the left graph were getting less sleep than the animals on the aspen chip bedding over the same time period. So that was enough for us to go, wow, this is affecting not only this model, it also affected a pain model that we were doing some research in, and we had to go back to the aspen chip bedding, and we, as an IACUC, decided to make the guideline that we would not use corncob bedding in our facility based on this scientific data that it was really causing problems for several different models. So keep that in mind. When you start looking at temperature and lighting, something as simple as, you know, the lighting can and the temperature, they can affect the physiology. And so we've had several instances where we have aged animal models, and when they start getting older and they're, you know, starting to have some old age issues, you know, maybe their renal function isn't as optimal, just like older people, you tend to start having more, you know, cold temperature stress and feeling the cold, you know, in your body a little bit more. So what we've noticed is that if we just increase the room temperature just a little bit for those rooms that have aged animals, like aged rodents in particular, it can make a real difference to them feeling a little bit more comfortable based on behavior that we can assess, and maybe, you know, decrease the problems that we see. I think many of you have probably seen this article from 2013 about individually ventilated cages and how they can impose cold stress on lab mice. So it's certainly something for us to keep in mind. And then this other publication in 2013 by Brianna Gaskell was about the impact of nesting material on mouse body temperature. And so making sure that we're giving enough nesting material so that the mice can build good nests and they can find warmth inside those nests and they can maintain their physiology a little bit more easily has really been shown to be a very positive thing for the animals who do that nesting in the natural environment. Then we look at lighting. So obviously we all know that photoperiod is a critical regulator of reproductive behavior in many animal species. And so inadvertent light exposure during the dark cycle has to be minimized or avoided. There was this really good article in 2011 about eliminating animal facility light at night, or they called it LAN, and its effects on the circadian regulation of rodent physiology, tumor growth, and metabolism. And so they were trying to relocate a cancer research lab from one place to another, and they had to really figure out how to minimize light effects on the tumor growth. And so again, a very good article to look at. Something as simple as some of these doors that we have on the animal rooms that have a window that can allow light in. The other thing that they showed in this article is that a leaky door jamb, that maybe our doors are not sealed all the way around, and so light from the hallway may be getting in and affecting some of these rodent circadian rhythms. And so again, checking the lighting system. Sometimes we rely so heavily on our computerized methods of monitoring things, and we never double check that they're actually working to our specifications. So making sure that once in a while we come in at night and make sure that really things are happening the way they appear to be happening is a really good practice. So some of the husbandry considerations, just to review them again, has your staff considered the impact of diet on animal models? And if you're, are you using soy-based diets? And if so, you might want to consider how that might be affecting the drug levels if you're dosing any of animals with pharmaceutical agents and measuring those levels. Really carefully consider that bedding, because it really was a very eye-opening experience for us when we had the corn cob issue. And then just the nesting material, making sure you're consistently giving enough so they can build those really complex nests. I know it's a cost factor, but again, in the end, if you're optimizing the welfare of the animals and stabilizing their physiology and their ability to maintain that thermoneutral zone, you're going to have better science coming from those animals. And then just checking the lighting to make sure it's not exposing these animals to unnecessary light at night. Behavioral enrichment. So we recently had the great luxury of hiring a full-time behaviorist to come and work with us at AbbVie. She started just a couple of months ago. We're thrilled to have her. She helped me with these next two slides. And what she wanted me to emphasize for all of you is that, well-conceived enrichment provides animals with choices, okay, and a degree of control over their environment, which allows them to better cope with the stressors in their environment. And that comes directly from the guide. And that successful programs for enrichment should incorporate multiple tiers or layers of enrichment so that you facilitate species-specific and species-appropriate behaviors. So it's more than just throwing a toy in a cage or giving a treat, but allowing them to have some control. So well-facilitated enrichment programs aim to increase physical activity, stimulate natural behaviors, and prevent or reduce stereotypic behaviors. Something as simple as what we've done in our facility for rodents, rats love to gnaw. So we have these wooden blocks. You'll see the wooden block in the center. And we did a study, a side-by study, to compare rats on the wood blocks versus rats without the wood blocks, and did it affect the GLP absorption of some of the compounds that we were giving the rats, and we found it didn't, and so we were able to publish on that. We've also used popsicle sticks, sterilized popsicle sticks, for the rats to chew on. And obviously mice like to build nests, and obviously the one thing that we all try to do is the social housing, because all the animals are social to some degree. Rabbits are the ones that are a little bit of an outlier, but most of the other species get along quite well, other than male mice. This is a picture of something I'm really proud of, and this is the dog socialization and enrichment program that we've established at AbbVie. We actually just celebrated our 5-year anniversary for this program, and basically what we did was we developed a program that we invited employees that are not members of the research team, they're not members of the animal care staff or the vet staff, but members of the company that are like, secretaries, lawyers, public relations staff, safety personnel, all these different areas of the company. We let them know that if they wanted to come and enrich the lives of our dogs and be enriched, that they could be trained to participate in this program. When I started this program 5 years ago, I was very nervous. I'll tell you very honestly, I was nervous about it. I wasn't sure if it was the right thing to do, but in the end, I kept thinking there are a lot of dog lovers out there who would love to do this, and so we did do it, and I got support from senior management to open the doors and get them trained and let them in, and I can tell you it's been so much more successful than I ever dreamed, and we have new volunteers all the time. And we train them on how to, go into the facility, and we assign dogs to them, and they take them to this playroom. And so we basically took one room and painted it and added a little play area so that the volunteers can take the dogs to this room. And it's been a great thing. So I highly encourage you to consider something like this. Very innovative. So some of the behavioral enrichment considerations: do you have a coordinator? I know it's a luxury. I know many of you can't, justify hiring a full-time person, but maybe something as simple as an enrichment committee. That's another thing we established, is an enrichment committee. So you find the people who are really passionate about optimizing enrichment, bring them together, have them meet regularly, have them establish some standards about what enrichment you're going to give the different species, and be consistent, and always stay up with the scientific literature, because it's very important that we only give enrichment that is scientifically validated to be a real enrichment and a benefit to the animals. We shouldn't be throwing things in there. An example, for a while there were people giving marbles to the mice. That's actually a model for stress in neurobehavioral research. And when we realized that, we quickly stopped giving marbles to the mice. So there are things we have to really be careful about, and just this is a list for you to review at a later time if you're thinking about how to expand your enrichment program. Training. So this is another area that I know many of us are trying to do the best we can with training, but it's a hard area to get your arms around. I know it's certainly an area that we struggle with, and because we do not yet have a full-time training coordinator, and we recognize that as a gap. So I'll challenge all of you to consider whether you have a full-time training coordinator. You know, is your program of an adequate size to justify that? Do you have good training SOPs? I think that's a really critical part. I can say that we have very good SOPs for training. We just have a struggle managing the bigger program because we don't have a full-time person dedicated to it. Do you have hands-on training for common restraint, blood collection, dosing, and humane euthanasia? I think those 4 things are very, very important to provide hands-on training when possible. Because again, if you don't do it right, that's where a lot of the welfare problems arise. Proficiency. So our IACUC has started asking for real-life proficiency, where someone has to do the technique in front of someone who knows, you know, what they're doing, and they have to verify they do know how to euthanize humanely. They do know how to do cardiac blood collection in mice humanely. They make sure the mice and the rats are fully anesthetized before they try to take the blood from the heart stick. Doing a perfusion, a cut down or an abdominal incision, and doing a perfusion, obviously if you're not validating that that animal is fully anesthetized, you can cause a lot of pain and distress. So we always do proficiency observations in some of these more challenging techniques that are very commonly done in many of our programs. And then just remember training documentation. I know it's hard, but if it's not documented, it didn't happen in many ways, right? There's no proof of it. So look at your training documentation and consider ways to optimize that. With handling and restraint, obviously we always try to minimize the restraint to reduce stress on the animals. I tried to do as good a job as I could with my limited graphic design skills. We had some scientists working in the cancer research with mice where they have to do a lot of IV dosing of either cancer cells or cancer drugs, potential chemotherapeutics. And so they came up with a very simple restraint device that is very minimal restraint. And it's a metal platform with a metal 90-degree angle back. And in the back of the 90-degree vertical piece of the metal, there's a hole. And so they literally bring the mouse to the metal platform, and they stick the tail through the back of that vertical piece, and that way the mouse is totally unrestrained on the other side of that metal piece, and they can easily get the IV injection done. I thought it was a really smart way to do it. We probably should publish it because it's just so simple. But I would, again, be innovative in making these restraint devices that work for you and your program. And something as simple as just making sure the lighting is good. This tail illuminator restrainer I thought was a really cool thing, just to help make sure that vessel is really easy to see since they're very tiny. Some of the techniques to consider refining. So with blood collection, something as simple as a tail tip prick. So if you know, I know in the industry, many people still use a snip of the tail tip with some sharp scissors for rats that are being used for diabetes research or mice that are being used for diabetes research. For the larger species in particular, rats, you can use a needle and just prick the tip of the tail and still get a blood glucose sample without having to snip off the tip of the tail. It's less invasive, less painful, quick and easy. It works great. We've certainly done it here. The other thing is microsampling. So this is something that in the pharmaceutical industry is very impactful, but I'm sure it can be impactful elsewhere, as our equipment to measure different analytes in the blood, CBCs, gets more sophisticated, and we don't need as much blood volume as we used to. We could possibly reduce the amount of blood volume needed by double checking with the people who manage those labs, saying what is the minimum volume of blood you need, so that we can reduce how much blood we have to collect and possibly refine the way we manage these animals. So we were able, with a group of mice and the PK sampling that we do, pharmacokinetic sampling we do repeatedly, we reduced the number of mice from 33 down to 3 by using microsampling and getting much smaller blood volumes for every time point, and we could get a whole curve in 3 mice and have better scientific data and better and less animals and much more efficient process. With dosing, using flexible dosing gavage needles or tubes can be obviously less traumatic than the metal gavage. Now, some people strongly prefer metal gavage tubes because of the ball on the end, making sure you never go into the trachea. But for the plastic gavage tubes can be quite useful, and some people do prefer them because they're not quite as rigid and not as likely to cause trauma. I liked this website here, procedureswithcare.org.uk. I would encourage you guys to go there and check out, even they use a soft red rubber flexible tube for oral gavaging in the rat. I've never done that, but it certainly looked like it was something worth considering if you had maybe a smaller study to manage. Surgical techniques. How do we refine these techniques to optimize animal welfare? So veterinary oversight of all surgical animals is what we're able to do in our company. Our company is a mid-sized animal program here in Lake County. I know this isn't feasible for some of the really large academic programs, but I would certainly encourage you, especially with some of the rodent surgical models that are more invasive. What we did when we were first getting our arms around this issue is we started asking the research staff that we made it mandatory that they would notify the vet staff when surgeries were being done. So at least we would know where those animals were and how many and what the kind of surgery was. And then we would then go and our vet techs would take a look at those animals, and especially if they're doing it on a Thursday and we want to monitor those animals over the weekend. That also helps us to stay on top of surgical clip and suture removal and provide supportive care like sub-Q fluids or analgesics if needed. So, mandatory proficiency demonstration of aseptic technique and tissue handling is something we require in our program. I think many of you do too, but not all programs do require that. That is a really good way to at least make sure that everybody's on the same page with aseptic technique and tissue handling. As far as analgesia use, one of the areas that, again, I think we don't have enough publications on in the literature, but it's getting better, is analgesia use. So we faced an issue here where there was real concern by our cancer scientists that if we gave 1 or 2 doses of buprenorphine, that their models of orthotopic cancer research would be affected in a negative way and that they wouldn't be able to get the data they needed. So we were able to convince them and work with them and partner with them to do some side-by-side studies. And with the side-by-side studies, we were able to show that 1 or 2 doses of buprenorphine did not affect tumor growth in this strain of mice with this tumor cell line. We then brought this data in front of the IACUC and said, based on this information, we feel that it is adequate to give analgesia to all the tumor mice that have to go through a major abdominal procedure, and that was able to get implemented. Likewise, the charts on this table, we had an IL-23 inflammatory ear injection. So they would inject the IL-23 in the ear of the mice, causing swelling of the ear, inflammation, and then they would dose the mice with anti-inflammatories to see if they could reduce the swelling of the ear. But they had to do some kind of a mini pump surgery earlier on, and so we challenged them and said, well, we want you to give analgesic for the surgical procedure. And they were concerned at first because they're studying a lot of anti-inflammatories, and since our analgesics can often be anti-inflammatory, they were worried about the effect on their data. So they went ahead and did the study, and they showed, again, that 1 or 2 doses of buprenorphine, there was no effect when you did that 48 hours before the IL-23 injection. So then we were able to implement analgesia for those animals in studies that prior to that we hadn't gone that route because of their concern. So the science is really important to get it out there and to really get the data to prove and to reassure your scientists that things are going to be okay. As far as animal health observations, we have language that says veterinarian or veterinary technical staff must be involved in observations of animal models. I've already mentioned this with surgical models, but also with inflammation models, with cancer models, with infectious disease models, some of the more challenging models. EAE, what is it, allergic encephalomyelitis, that they have ascending paralysis in the rats. Those are really challenging models, and you need to make sure your veterinarians are partnering with the research staff in providing the best oversight and minimizing unnecessary pain and distress. Regarding supportive care to minimize harms, again, just making sure that the staff have the expertise with the species. Hydration is so critically important, and there's so many options now, water-based gels and liquids that can have nutrients added to them. Medications can be added to these gels for treating pinworms. Any of these things can reduce stress because less handling, less stress, better supportive care. And obviously, I don't know if any of you have ever been dehydrated. I've been dehydrated. It doesn't feel good. You don't feel good at all. So we have to really maintain their hydration well. Weight monitoring obviously is critically important, providing nutritional supplements. One of the things that I saw on a recent study where we had some real challenges with multiple inflammatory diseases is first we were just monitoring weight loss from the beginning of the study throughout the progression. But when I started thinking about it, they were young mice and they were still in the growth phase. And so when I started comparing their growth rate to what the normal controls on that study were, they were even more debilitated when you looked at their lack of growth. So I think something to think about, not just body weight loss compared to their original weight, but are they gaining appropriately for the age of the animal and the age that they are and the species and strain that they are. Temperature monitoring using those sub-Q chips or some other refined ways to take temperature. I don't know if any of you have used, I was just at the doctor last week and they took it just again with on the forehead. I know that there's more and more research to look at taking temperatures by using some of those more advanced thermometers that are less invasive, but I haven't kept up with all of that. So again, something to think about for little refinements that we could do later on. And I think, again, I'm going to make this comment with the picture at the bottom. We must ensure consistent welfare standards for all species, no matter how small, because I have to say that in my 20-plus years in the industry, I definitely think at times there is more care given for the larger species than for the smaller species, not because we don't care, but because maybe we're not as good at assessing the mice or the rats as we are with the dogs and the monkeys and the larger animals. So just, again, something that we need to challenge ourselves to continually get better at is really providing that supportive care for the rodents at all times. Humane endpoints, obviously, this is always a challenge. When is enough enough? Having those pre-study meetings with the research staff to agree on humane endpoints, I think, is the best way to manage this proactively. Agreeing that a certain body weight loss or a combination of things is when we're all going to say this is enough. The scoring sheets are very useful. Now, they're impractical if it's a huge study. They're impractical if you think you have to start it from the first day of the study all the way through. So I think, again, be rational when using scoring sheets. Start using them when it becomes challenging to make those humane endpoint decisions. So if your animals start getting more debilitated, 3 weeks into the study, and that's when you're really questioning, is it an endpoint and isn't it? Those scoring sheets really help you to be objective and be more consistent in applying those humane standards. I wanted to show this example. This is an Excel scoring sheet that our vet staff recently used on one of the more challenging mouse inflammatory models. And the cool thing is, it's a little bit hard to see, but the way they developed this Excel scoring sheet is that when the mice would lose a certain amount of weight, you could see the pink bar, 20% loss. It would highlight and flag the mice that had lost 20% of their body weight. So there are ways to make these kind of automated to be more user-friendly, and you have to spend less time adding and subtracting yourself because the Excel spreadsheet will do it for you. So keep that in mind, and if you guys would like a copy of this, I'm certainly willing to share it with you. Let's see. So I just was highlighting body weight loss and then body weight suppression. So we were flagging both of those with this study. So I don't know how many of you are from pharma companies, but with tox studies, we certainly are starting to try and make sure that we consider staggering the dosing for some of the more challenging studies. So for example, instead of dosing low dose, middle dose, high dose all in the same day, if we don't have enough information about the compound to be dosed, we will ask them to dose the low dose and the middle dose on the first day. And with things like antibody drug conjugates, which you can have adverse effects about 2 weeks later or within the first 2 weeks, you may not want to dose the high dose monkeys until 2 weeks later, so that you really know what's going to happen in that middle dose group and that low dose group. And if you don't need to go to the high dose because you see more toxicity in the middle dose, then you've really prevented some unnecessary pain and distress. The other thing that we have done at this company is we create a form for each tox study to make sure that we know exactly what the likely potential toxicity is, what organ is likely going to be affected by this class of compound, and have those vets, again, ready to intervene if there's likely to be seizures because this is a CNS active drug, if there's likely to be emesis or diarrhea because this is a cancer compound that's going to cause some GI upset and sloughing. We need to be on top of that and ready to intervene at a moment's notice. As far as IACUC and ethics committee oversight, just making sure you have those clear guidelines that you can quickly refer back to minimize the harms, to guide how much the dose volume is for the different species. Again, clear training and proficiency expectations. Adverse events. I hope all of you, if you're involved with an IACUC, that you're getting regular updates from the vet staff and the whole program about unexpected complications that occur with the different animals. Because again, you may end up seeing as an IACUC that there are some trends in repeat issues that need to be addressed. And something as simple as we were having dogs that were having allergic reactions, and we needed to have a guideline to say how were we going to manage those allergic reactions in a consistent way no matter when they occurred. It's all about creating that culture of animal welfare and having refresher training to emphasize the importance of animal welfare and the 3 R's, and enhancements to our practices, that we're never satisfied with good enough. We want to be the best. We want to optimize, and we're always looking for ways to improve our oversight and care of the animals. Something as simple as making sure you have well-defined action levels for noncompliance with IACUC procedures and animal care and use procedures. Not to be, like compliance police, but if you have investigators who are pushing back on optimizing welfare and doing things that really are what you expect, then you have to have a consistent way of dealing with some of those issues. And so again, we've created minor, moderate, and serious noncompliance levels, and how we interact with them are different levels of, action. Some are just an alert, some you get a letter of warning to say, we're really concerned about, some of the ways you've managed this, and some we put them on probation, so don't be afraid to do that. So again, building that culture of welfare, you have to be proactive, you have to be innovative. We have the great blessing of having a full-time 3Rs coordinator. Again, I know it's a luxury that many of us can't have. We've created an alternatives committee, and that alternatives committee sets annual goals to look at different 3Rs options that we'd like to maybe proactively promote across the company, across the program. We'll do training initiatives, things like that. And that's all on a volunteer basis, so you don't have to have a full-time person to do that. I already mentioned the dog socialization. We adopt out our beagles whenever we can. We have a global enrichment committee, so we're consistent. We have an animal welfare committee that really is a high-level committee to establish company policy. And then we do recognize employees by having an award program. Employees who are advancing the 3Rs, who are going above and beyond, we want to recognize them and give them awards and really make that well known. So this is Natalie Bratcher, 3Rs coordinator, and again, I'm very proud of Abby being, well, we were Abbott when we brought her in as a full-time 3Rs person, but again, we're very proud of the fact that she is able to help coordinate and implement 3Rs for our company. And I already mentioned the Alternatives Committee, annual goals, 3Rs Journal Club. The Journal Club can be really useful because it is a brainstorming time, and we actually started a zebrafish lab after a Journal Club review of a zebrafish article. We realized that we had a gap and that we should try to fill it with a zebrafish lab, and it's really taken off well, and it's, again, something we're very proud of. And I already mentioned this. We actually have 3 levels to our award program. The first level, we just give out a lunch pass, less $5 lunch pass. I don't know if all the academic institutions can do that, but if you can, it goes far in helping people to get motivated to continue to look for ways to advance welfare and the 3Rs. We have a second level giving up to $50 gift certificates, and then we have a third level award plaque, a luncheon. We ask them to present on their 3Rs implementation, and we highlight the advancement, the 3Rs success story in an article across the company. If you want to consider creating an award program at your institution, please go to this JLAS article from 2015. I was the lead author on this, and it gives you kind of some of the things to consider in creating an award program for your own institution. The reality about the present state of the 3Rs is that for the foreseeable future, drug development and safety testing will continue to depend on animal models, right? Because even though there are many in vitro systems that are starting to get validated, they don't completely mimic the complexity of the higher organisms. So, will we ever get to a point where 3D organ systems can replace animal use? I have my doubts. I hope. It would be great to see that happen. Not so sure that'll ever happen in my lifetime. But the reality is humane animal testing is still required and will continue to be required for many, many years to come. And we just have to really do our best to minimize the pain and distress and to do what we can so we can very much educate the public and talk about our commitment to ethical science and research. So, just a quick summary. We talked about receipt and acclamation procedures, about the environment and all the different things to consider there, about training, about the techniques we use, about providing supportive care and humane endpoints, about our oversight procedures, and about that culture of care. The reality is there's so many things we can do to tweak our program, and I feel like most of us know that we're never quite perfect. I would love to see a perfect program. I have yet to see one unless it's like a one-room animal facility. But the reality is most of us just need to remember, we really can enhance the science through small changes in the way we do things, making sure we partner with the research scientists and do everything we can to enhance the work and the welfare of those animals so that we really can discover better medicines, better understanding of science and the way we live. So I guess what I will do at this time is I will open it up for questions and see if anybody has any questions.
Excellent presentation, Dr. Medina. Thanks for bringing that information to us. So before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window, and we'll get to as many as we can. So our first question comes from Gregory at Medtronic, and he asks, if volunteers are used to calm animals such as dogs that have been received, is this documented in the facility records, and are the volunteers then considered to have taken part in a GLP study if the animals are subsequently used in a GLP study?
And I have to... Okay, I started talking before I had full control. So that's a good question. To my knowledge, and I may be wrong, and I'll have to look into it, to my knowledge, when we receive the dogs, they are not yet put on a GLP study at that point. So we have a protocol for receiving animals. And we do that purposely because we don't assign every dog that we order onto the GLP study. We actually do the physical exams and make sure that we pick the dogs that we think are the best suited for the GLP study, and we typically leave a couple dogs out. So at that point, they are not considered to be on the study until they have been transferred to the protocol, the GLP protocol, and then, so it's really a non-issue at our facility, but maybe you manage it differently, and maybe you want to consider creating a protocol for receipt of animals so it's not an issue for you. I don't know.
Daniela from the Federal University of Sao Paulo asks, how could the IACUC help an investigator to decide which outcome to look for to not have to give up analgesia?
So I'm not sure, I think I understand your question. So the two examples that I gave, we had cancer researchers that were nervous about tumor growth. So they were afraid that if you gave an analgesic, and they had some literature to show why they were concerned. And the literature was older literature, and it was higher doses of what compared to what we wanted to give for pain relief. So when they made the argument that this scientific literature proved that they couldn't give analgesics, what we said as an IACUC is, well, no, we're asking to give a lower pain relief dose, not a high repeat dose like in this literature source. And we don't see any literature that addresses specifically what we're concerned about. So we said, we want to do the study with you and prove scientifically one way or the other, does 1 or 2 doses of buprenorphine at the pain relief dose affect your tumor growth or not? And so we worked with them, partnered with them to understand their concern, and then we told them what dose we wanted to use, and that's how the study showed us that the buprenorphine was not going to impact their research. The second example I gave was about an inflammation model. And so in that model, the mice were injected into the ear with IL-23 to cause inflammation. And again, what they were concerned is if we gave buprenorphine at an analgesic dose, because they had to do a surgical procedure to place a mini pump, they were afraid the buprenorphine would affect the swelling of the ear 2 days later. So we said, well, we don't have any proof. There's no scientific literature to support that concern. Get the data and show it to us. So they did the study, and they did it within a week. They did it very quickly, And they came back, they showed us the data, and they said, you know what, you're right. There is no effect on the swelling of the ear. We can give pain relief, and we will adopt that recommendation. So it really is the investigator's responsibility to do the study, but the IACUC, or the ethics committee, has to have the firm stance that that is the requirement. So one of the things that we did here recently, about 8 months ago, we established a policy that made it very clear that analgesia is the default expectation for all survival surgeries or invasive procedures, and it is the investigator's responsibility to do side-by-side studies if they are concerned that it would impact their research. And so again, that is what we're asked to do as a committee, is to set the standard and then hold them to it. I hope that's helpful.
Agnieszka from the Institute of Industrial Organic Chemistry asks, is there any guide or rules about dosage selection in repeat dose toxicological studies, knowing, for example, only the LD50 of the test item?
So again, okay, so I'm not sure I can answer that question fully, but what I will tell you is I'm part of a consortium right now, a 3Rs leadership group consortium. And one of the things that we're doing right now is we're looking at developing a guideline on maximum dose volumes for different species. Now, it doesn't necessarily take into consideration what you're asking, which is the repeat dose and the LD50 information. So that's a bit more challenging. I'm going to pull up a reference. I don't know if you can see this. Let's see if I can get this right. The Toxicologist's Pocket Handbook. This is a good reference to consider if you don't have it. Michael Derilanko is the author. It does have some information in that talks a little bit about repeat dose concerns. Other than that, I saw a reference today, and I want to say it literally was just published in 2016, so hot off the presses. And it is dealing with, it's an 82-page reference from, I want to say, I could share it with the group later maybe. If you leave your email on the, with the group, I could possibly send you the reference information for this guideline. It basically is dealing with excipients and vehicles and the toxicity that can be seen when we do dosing in the variety of animals. And so I don't know if that would help you out, but there's certainly a lot of information that we need to start sharing better. And so I'm certainly willing to kind of hand that off if we gather your information on how to contact you.
Great. Okay, we're going to give people just another minute. They're formulating some questions. Give you a minute to submit them. Oh, yes. Okay, hang on just a minute here. Okay, someone is provided some information, not a question. Okay, so if there are no more questions, I would like to once again thank Dr. Medina for her presentation. Do you have any final comments for us today, Dr. Medina?
Sure. So my final comment is just again, you know, thank you for participating in this web training today, and just remember that again, everything we do is about you know, advancing science and medicine and making sure that those animals are treated ethically. And we have such a responsibility to do the best job possible. And we need to share information better. Please, I encourage you all to, go to those national meetings, the ALAS meetings or the FALASA meetings, whatever meetings you go to on the topic of animal research, and share your best practices and publish and become those leaders to advance animal welfare and science so that the public that has concerns about what we do when we work with animals can feel reassured that we're doing it the best way we can. So thank you so much for your time.
Thank you, Dr. Medina. This is an educational webinar and thus offers free RACE continuing education credits. After the webinar is over, and to get your CE credits, click on the CE button located in the bottom left-hand corner of your web page. Today's webcast will be available for on-demand viewing through August 4th, 2016. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You are welcome to forward this announcement to any colleagues who weren't able to join us today. Thanks for logging on and participating in today's broadcast. We'll see you next time.